Welcome back friends. Welcome back in another video tutorial from microbiology with Shagnik. So in the previous video we have talked about how we can cleave the peptide bonds, right? But for denaturing, totally denaturing a protein, just only cleaving the peptide bond is not enough, because there are various type of bonds available that joins two peptide chains together, right? That may be non-covalent bonds, that may be disulfide bonds, etc, etc. So today we will going to learn how we can break those non-covalent bonds as well as disulfide bonds. So let's get started with the video. Okay, so cleavage of the disulfide bonds. Now you may ask that why this kind of things is necessary, because in the very first video of this part specifically, that is protein sequencing and assay, we have discussed about the determination of amino acid composition, right? And to determine the amino acid composition, it is very important to degrade a protein totally into its very basic form, right? So that is why cleavage of those kind of non-covalent bonds as well as disulfide bonds is very important, right? Not only that, in case of SDS page kind of things, where we are trying to determining the molecular weight of the protein, where very important is to degrade the protein into its very basic form, right? So that is why this kind of things are very important. So first we will going to understand how we can break non-covalent bonds. So non-covalent bonds can be broken down by urea or guanidine hydrochloride. If you have watched my protein folding video, then there we have discussed about the Christian Anfinsen experiment, where we have discussed that he have denatured completely RNase A into its very basic form by using 6 molar, sorry, it was not 6 molar, 8 molar urea along with that beta mercaptoethanol, right? So now you can understand why we have studied that he have used urea, right? He have used urea to break those non-covalent bonds. Along with that, guanidine hydrochloride can be also used, but urea, especially 8 molar urea, is very important. Okay, now we will going to learn how we can break disulfide bonds. So for breaking disulfide bonds, that means this kind of things, there are two methods, two common methods are available. One is oxidation, okay, another one is obviously reduction. Okay, so oxidation of disulfide bond by performic acid is very important to break the disulfide bond, and it will give you 2 cystic acid with SO3 minus side chain. Okay, and now try to think about this. If you have 2 cystic acid with 2 SO3 minus side chain, as they are same in charge, they will repel each other. That is why there is no chance of disulfide bond reformation. Okay, because remember, when we are dealing with breakage of disulfide bond, just breaking the disulfide bond will not going to solve our problem, because after breaking there may be a potential chance that this disulfide bond can be regenerated spontaneously, right? That is why we have to ensure that they cannot regenerate, right? Otherwise our entire effort will be wasted. So it is very important. So we can use the oxidation process by using performic acid to break the disulfide bond. Okay, so now another important method, that is the reduction, and this reduction can be done with the help of beta mercaptoethanol. You have probably heard about this beta mercaptoethanol earlier, along with that dithiothreatol, and we are using this reduction process in case of our SDS page, right? And this dithiothreatol things is also known as Cleland reagent. Okay, so remember, this type of reduction will give us 2 cysteine residue, that means cysteine with SH at the side chain. Okay, and if two SH groups are in close proximity, then there is a possible chance of formation of disulfide bond. Okay, that is why reduction process includes a one extra step where we have to modify these SH groups in such a way that they cannot recreate disulfide bond, and we will do this with the help of acetylation process by iodoacetate. Okay, so that is the idea of how you can break the disulfide bond. So two process, one is oxidation, where it is a one and only process, you add performic acid and it will give you 2 citric acid with SO3 minus group, and SO3 minus group together cannot form disulfide bond, so you are okay with it. But when we are using reduction, for example beta mercaptoethanol, for example dithiothrietol, it will only convert them into cysteine, right, with SH side chain. So this SH side chain can be regenerated into disulfide bond. So for protecting this, we will perform acetylation by iodoacetate so that this SH bond cannot be regenerated. Okay, so that is the idea of how we can break the proteins non-covalent bond as well as disulfide bonds. Okay, so that is the idea. So now by using peptidase you can break the peptide bonds. Urea and guanidine hydrochloride you can break the your non-covalent bonds, and then by beta mercaptoethanol dithiothrietol along with iodoacetate, or by using only performic acid you can break the disulfide bonds, right? And they are the major type of bonds you may find in most of the proteins, right? So now you have all the tools to break a protein into its very basic unit to analyze them properly, right? So that is the idea of our protein degradation. So now, not protein degradation is not a term to use, protein denaturation. Okay, so now after these two video, in the next video we will start to discuss about protein assay. We will going to talk about very important type of things. Okay, so stay tuned and thank you for listening to this class. Okay, so bye bye.